What is up, Bio people? Here we go again for another vlog, and for today's session, we'll be discussing what petroleum microbiology is, and also we'll be dealing with some applications under this topic. But before we go along with our discussion proper, allow us to introduce ourselves. I am Mary Rose Palomar.
I am John Noxel Eclipse.
I am speaker number 3.
I am Jacqueline P. De Los Santos.
And I'm speaker number 5.
Keeping you company for today's agenda. First off, what is petroleum microbiology? Petroleum microbiology, by definition, is a branch of microbiology that deals with the study of microorganisms that can metabolize or alter crude of refined petroleum products. Here are some general applications where this could be used, and that is for bioremediation. What about this bioremediation? Bioremediation of oil-contaminated soils, marine waters, and oily sludges is a feasible process of hydrocarbon-degrading microorganisms that are omnipresent and are able to degrade most compounds in a petroleum oil. And in some simplest cases, indigenous microbial communities can degrade petroleum where the spill occurs. But in more complicated cases, various methods such as adding nutrients, air, or exogenous microorganisms in a contaminated sites can be applied. For example, bioreactors involve both of the natural and the additional microorganisms in a controlled growth, and so it will yield to high biodegradation rates in a wide range of media.
Not only it is used for bioremediating, it can also be applied for commercial purposes. Two well-known oil spills exemplify large-scale marine bioremediation applications. In 1989, the Exxon Valdez ran aground spilling 41.6 billion liters of crude oil and launching one of the first major bioremediation efforts for an oil spill. Cleanup of Alaskan shorelines rely in part on fertilizer application to augment bacterial growth. In 2010, the BP Deepwater Horizon oil spill released 779 million liters of oil into the Gulf of Mexico. So, this was the largest oil spill of all time, and indigenous petroleum microorganisms played a major role in petroleum cleanup and degradation. Another model microorganism that is studied for its role in bioremediation of oil spill sites and hydrocarbon catabolism is the alpha proteobacteria alcanivorax. Alcanivorax burkimensis degrades alphatic alkenes through various metabolic activities. Alcanivorax burkimensis utilizes linear hydrocarbon chains in petroleum at its primary energy source under aerobic conditions. When further supplied with sufficient limiting nutrients, it grows and produces surfactants glucolipids to help reduce water tension and enhance hydrocarbon uptake.
Aside from all the applicants mentioned earlier, another important thing to remember is that this could be used as biosurfactants. These are microbial synthesis surface active substances that allow for more efficient microbial biodegradation of hydrocarbon in bioremediation processes. And there are two ways by which biosurfactants are involved. First is the increase of the surface area of hydrophobic water-insoluble substrates. Growth of microbes on hydrocarbons can be limited by an available surface area of the water-oil interface. The second one is the increase of bioavailability of hydrophobic water-insoluble substrates. Biosurfactants can be enhance the availability of bound substrates by disturbing them from surfaces or means of increasing their apparent solubility.
So for the next one, I'm going to discuss the oil recovery. Microbial Enhanced Oil Recovery, or MAOR, is a technology in which microbial environments are manipulated to enhance oil recovery. Nutrients are being injected into porous media, and endogenous or added microbes promote growth and generate products that mobilizes oil into producing wells. The two general strategies in enhancing oil recovery are altering the surface properties of the interface and using bioclogging to change the flow behavior. Biomass, biosurfactants, biopolymers, solvent acids, and gases are some of the products that are added to the oil reservoir to enhance recovery.
The
fifth function points out the application of biosensors. Microbial biosensors identify and quantify target compounds of interest through interactions with microbes. For example, bacteria. The biosensor system may simply use bacterial growth as a pollutant indicator. Many analytical techniques require expensive treatment of oil samples and expensive equipment to detect the presence of pollutants. Bacterial biosensor systems offer the potential for cheap, robust detection systems that are selective and highly sensitive.
There you have it, BioTips. That is pretty much everything for our vlog today.